I'll just start with the first question. Obviously, both of you have received a number of awards, and this is sort of the glory days of mRNA. But I'm interested in hearing about the more sort of challenging days when maybe nobody wanted to invite you to give a talk, or nobody thought that there was any promise in what both of you were working on, and maybe some government grants that told you were crazy and we're not going to give you any money. So, Dr. Weissman, do you want to start and tell us?
Oh, you can.
Well, either one.
I can say that Drew is usually very very quiet. We are very different people, and but that time when we had our publication ready and will be published, that was the first time he said that, listen, tomorrow the telephone will ring, the people get excited with our results. So, of course, it never happened. We were not busy picking up the phone. We went back to be busy in the laboratory. But, he was very optimistic on that. And we had to wait another 10 years or so that somebody is referencing our publication. And then they discovered that we did that.
So I'll tell you stories of what it was like going to conferences during those years. And I trained in Tony Fauci's lab, so my lab did mostly HIV work. And I would go to HIV conferences, and I would sit down and I would talk to the leaders in the field, people like Tony Fauci or Gary Nabel or others. And I would spend time telling them about the RNA work that we were doing. And they would sit there and smile and nod. And then they would turn and honest people, they would say, I think you're wasting your time. Why don't you do something useful with your life? Why don't you investigate something worthwhile? And I would try to explain to them why I thought RNA was worthwhile, but they were gone at that point. So I'm a quiet person. I haven't gone back to Tony and said, by the way, do you remember when you said...
But usually when we said somebody that we are working with RNA or mRNA, they really felt sorry for us. Because, if any of you ever work with RNA, that's one thing that it degrades so quickly.
Some questions? Yes, go ahead.
Everybody in the audience has, of course, heard of mRNA, but I don't think all of us are aware of the incredible scientific advances that have led to this. And I can't expect you to give an hour lecture, but I'm hoping maybe you could tell us, those of us who don't know, some of the incredible things that have happened that let you do what you're doing.
I might say about that, so the RNA was discovered more than 60 years ago, and I think that in every 2 decades something significant happened. So discovery, and then 20 years, people try to figure out how it looks, the different structural elements in it. And then in 1984, first time we could do RNA in the tube, because that enzyme, the T7 RNA, SP6 RNA polymerase, which is a phage polymerase, is discovered. So we could make in a tube, and that's actually T7 was used for all of the vaccine if you get RNA vaccine. And then next 20 years, people used in the animal studies try to figure out what it is used for, and then establishing the first RNA company 2000, that was CureVac, and then the human trial started. So this was the progress, how I see it.
So, what I like to do is look at the future and what the potential holds. So the vaccines have had phenomenal success. They've been given to over 1 billion people. The efficacies in the clinical trials were nearly 95%, which is completely unexpected. But from that has now born, there's over 150 clinical trials for new vaccines. There's over 150 of diseases, including HIV, hepatitis C, malaria, tuberculosis. I can't really count how many different pathogens are being done right now. But beyond that, clinical trials for genetic therapies, for protein replacements, there's already been a phase 1 trial that has cured all of the high-dose patients of transthyretin amyloidosis, which is a disease that typically kills people 4, 5, 6 years after diagnosis. Those people are now cured with a single treatment with RNA. So the potential future to treat so many different diseases is really enormous.
Oh, yes. Good thing I looked up. Yeah. Why don't we start over here? Oh, yeah, we'll wait for 1 second. Well, while we're waiting for the mic, and maybe you don't have to answer this, were there ever any disagreements, scientific or otherwise?
Yes. Drew used to describe that, you know, Kati, from A to B, you get like me. Yeah. No, the science, you know, that was, you know, we try to come up with ideas and other things, and we listen to each other.
I mean, I think it's problematic if two people agree completely on everything, because
that's what I tell my wife all the time.
That's a marriage, right?
Well, it's iterative, so of course you would disagree.
I got the mic.
Yes.
Martin Curran, Princeton. One aspect of your story that I find so interesting is that when COVID came, there were 2 small companies that developed the vaccines then and brought them to the market. Why 2 small companies that most people had never heard of before, when we all know the big pharmaceutical companies, and they didn't do it? Why did it happen in a small town in Germany, in Mainz, and where you work, and with Moderna?
Actually, I worked in Mainz, not with Moderna, but at BioNTech. And I might say that it didn't, you know, the BioNTech and Pfizer had an agreement 2018 to develop a messenger RNA-based vaccine for influenza. So 2 years before the pandemic happened, it was already, we started to work together. And all of the trials, selection, formulation, everything was worked out. And at end of 2019, it was already ready for clinical trial with the authorities. And the COVID vaccine is just different. The order of the nucleotides, the composition of the lipid, everything was the same. So that's, you already know that in how quickly, in 6 weeks already, we can get a new vaccine. So it was not, it was a big company was behind BioNTech. And behind Moderna was the government, but you could see that still it was very helpful to have the company with big experience, how to run trials, how to scale up, and that was Pfizer.
Thank you.
I mean, I think in general, big pharmaceutical companies isn't that interested in brand new, untested ideas. That they don't go out searching for new ideas coming out of academics. They want ideas that have been in phase 1 clinical trials and show the potential to work. So it took small biotech companies to take the risk and say, this looks interesting, let's give it a try. What was amazing was how quickly they went from small biotech companies to producers of 1 billion plus doses of vaccines.
Many people were not aware. Many people already run clinical trial for infectious disease vaccine with mRNA. So 2015, CureVac already ran trials for rabies virus vaccine, and Moderna did run for influenza, but it in Germany because the German authorities were familiar that how to handle the mRNA-based vaccines, thanks to BioNTech and CureVac.
Yeah, the scalability was unbelievable. Yes, thank you for passing the
Okay.
microphone.
Leslie Aiello, University College London. This is definitely not my field. And I was wondering if you could explain why RNA vaccine is so revolutionary and so effective.
So the vaccine has two main mechanisms of action that make it different from others. For a vaccine to work well, in the old days, live viral vaccines were always the best. The FDA hated them, but they worked the best. And that's because they infect cells, they produce protein for a long time. The immune system needs constant loading of protein to make a good immune response. All of our standard dead vaccines, you get a bolus of protein that's gone in a few hours. So the RNA is a hybrid of that. The RNA makes protein for up to 10 days. The second thing, and this was pure luck, is that the lipid nanoparticle that delivers the RNA is actually a potent adjuvant that selectively stimulates B-cell responses. So those two things combined make the RNA vaccine extremely potent and effective.
Yes.
Annette Gordon-Reed, I would put myself in that same category. This is not my area of expertise. You mentioned an mRNA vaccine or dosage that cured a disease, people who have a certain disease. Will it ever develop so that when you get the mRNA, will it ever develop the vaccine that you don't get COVID again?
So
Turn off the recorders, please.
Yeah. The original vaccine that was made and tested, that people were getting the Wuhan strain, so the same strain that was in the vaccine, was 95% effective at blocking people from getting infected. So in the beginning, the vaccine prevented infection very effectively. The problem was COVID, like all RNA vaccines, mutates, changes very quickly. So the variants were much less sensitive and could not easily be neutralized by vaccine responses. The current Omicrons, the vaccine is about 10% effective at preventing new infection. What they did, and what was critical, is they made a cellular immune response that protects against severe disease and death. And that's why we still have 1,000 infections a day, but people aren't dying anymore because the vaccine gave them protective immunity that prevents bad disease and death.
Other questions? Yes.
Could you say something about the prospects and challenges for a universal coronavirus vaccine and whether there are serious chances that extending the mRNA approach would overcome them?
So it's a very long answer. We and multiple other groups are working on pan-coronavirus vaccines. And our goal is to prevent bat coronaviruses that have the potential to cross over. There are conserved regions across all the beta coronaviruses, which are the bat coronaviruses. And we're using those, and we and a few other groups have developed vaccines that protect against bat coronaviruses randomly selected. It probably needs a few more years of development because we want to include the MERS coronaviruses in that and the common cold coronaviruses to prevent all coronavirus transfer. I think it has potential. The data so far looks good.
You know, when you ask a question, I also always ask Drew the question of vaccinology. Should I take another vaccine or what should I do? So,
Well, wait a second. If you guys don't know.
So because all of a sudden everybody became a vaccine expert, and they asked me questions, but I said I have to also ask somebody. And that's also interesting because I learned all immunology from Drew.
Go ahead. Is there a microphone still? Oh, you have it. Oh, very good. Go ahead. Thanks.
Hi there, my name is Maura Sullivan. I'm a graduate student here. I'm doing my dissertation at APS. And I'm a little bit nervous to ask the question, but I'm interested, so I'm going to be brave. I heard a story about the inventor of a sewing machine needle that it came to him in a dream, the solution. So I guess I was wondering if you guys, and if not, totally fine, but if there is kind of some inspirations from like nature or other things that have helped you along the way with your work.
I mean, we did an experiment. I work in surgery department. We later became neurosurgery department, but I started at surgery department. And Drew, we really met at the Xerox machine, isn't it? That's true. That's true. And
What's a Xerox machine?
I like to brag, like my daughter here. I brag about that I work with RNA because I want to develop mRNA therapy for the stroke patient. And when Drew was interested in the vaccines, that measurement showed that it was RNA inflammatory. At that point, it seemed that for vaccine is still okay, but I thought that whatever I'm doing has to stop. But together we asked the scientific question that why it is this inflammation. Isn't that in all of our cells has mRNA and those are not inflammatory? And we came up with the idea that, okay, we will isolate RNA out of, not our cells, but cells, mammalian cells, different, and put on the mammalian immune cells to see that whether those are also cause inflammation. And then we were surprised that the tRNA, the transfer RNA, was not inflammatory. I mean, we didn't set out that let's find the non-inflammatory RNA. We just want to understand that. That's how we thought that the science leads us. And then we found that, it is not inflammatory. Could it be the modification, the nucleoside modification? And that came the next question. So we should test out, but how? You always face something that nobody ever done that. And we had to make nucleoside modified RNA. And the enzymes were not known how to introduce those modifications and so on. And so that when finally we could make nucleoside modified RNA, and we made 5 different ones, and more than 100 modifications is known, but we couldn't make those. And we found that one of them was, or 3 of them at least, was not inflammatory. Those uridine was modified, and one of them translated 10 times better. It was just, we went after science. So there were no dreaming. Probably we dreamed about things, but...
There were a few nightmares,
but actually, we worked hard, but we did not dream about that we have to modify it. It is just the science dictated it, isn't it?
Yeah, so it wasn't dreams, but it did often occur at 3 or 4 in the morning because we both have sleep disorders, and at 4 in the morning we'd be emailing each other back and forth talking about ideas.
And it was so good feeling that, Drew responded as I asked something at 3 o'clock, and I said, she's still up and working.
Thank God you both can't sleep. Yes, go ahead.
I wanted to ask, as someone new in science, new in their career, we're working in a time when scientific research is so siloed, and areas of interest are almost kept apart, divided by different conferences, different societies. I understand this is an ironic place to be asking that question, but I wanted to ask your thoughts. After a discovery like this that has such incredible implications across so many fields, how you think this can affect the future of how we research and how scientists collaborate and speak to each other across different fields.
So, I mean, I've never viewed science with that perspective. My lab currently works with about 250 labs around the world, collaborating on developing vaccines, therapies, and treatments. I've had a lab in Thailand, in Botswana for 20 plus years, where the goal is to transfer technology to those regions to give them access. I've built GMP production sites that are the required sites to make vaccines in Thailand, in South America, in Durham, North Carolina. That's not an LMIC, it's close. With the view that if you give people the access to the technology and collaborate with them, they can make drugs, vaccines, products for their populations that they need that big pharma may not have an interest in. So I think it's up to all scientists to be open-minded and to want to collaborate. My view of science is you get nowhere if you don't collaborate. If you're a single lab working on something by yourself and never talk to anybody, nobody finds out what you're doing and it doesn't go anywhere.
And for 10 years, I am participating in the mRNA Health Conference, where if somebody wants to use mRNA for therapy or they have a formulation or whatnot, small companies, students, it is a non-profit meeting, so people are coming to listen, people are coming who want to, like different countries, authorities that want to regulate or help to create those things, and people can present there that whether they have something to offer or they are seeking for GMP, mRNA, or something, so that there is a forum there. And if you would attend those meetings, you would not be surprised that the mRNA came out so quickly from a small company, because you already would aware of that clinical trial. Phase 2 clinical trial was running nucleoside-modified mRNA for heart failure, for example, and many other diseases.
Any more questions? Yes, go ahead.
I'm just curious if NIH wasn't funding your research, where were you getting your financing from?
Yes, so
I'm sorry, I'm going to repeat the question. If NIH wasn't funding your research, where were you getting your funds from?
So you can't repeat this and you can't tell NIH, but I came to Penn in '97 and got a decent startup package, but I had started writing grants before I got there. So I had an R01 when I moved to Penn, and I got a second one soon after. And I convinced the chair of medicine to let me keep my startup package. So we used that for about 10 years to fund the RNA research. It was just Katie and I sitting at a bench working together, because we had no excess funding to hire people. And it was, the two of us working together through all of this found the modified RNA.
We work in different buildings and different departments. I myself was hired in '89 by Penn, Elliot Barnett, and for cardiology, and he's just 8 days younger than me, and I convinced him that mRNA is the future. So I was a faculty position, a research assistant professor, who needs a green card, those get that. And so that's, he said, support the salary of mine, and, I get a grant. So after 7 years, I did not get R01, and then he left, and then I get to neurosurgery because one of the students in his lab by that time could convince the chairman of neurosurgery that neurosurgery needs a molecular biologist. But that was in
You never know.
97. And so I get a little lab space at neurosurgery and a little salary, which rose from 20 years from 40,000 up to 60,000 by 2010, but it didn't matter. And so I get a little salary there, laboratory, and that's how I could do things.
Okay, well, I'm going to wrap this up. It's an incredible story. Obviously, thank you for everything you've done for civilization.